Hello everyone, and welcome to my channel. I've been doing research the past 10 years on biomolecular condensates and the role they play in cancer metastasis, the origin of life, and quantum interactions in biology. This video, we're going to talk about the solution to the hard problem of consciousness. Imagine placing a droplet of dye into water. At first, it holds together like its own tiny world. Then it dissolves outward, fading into the surrounding liquid. Now imagine something strange happens. The dye suddenly begins to coalesce again. It pulls itself back together, it changes shape, it dissolves and reforms repeatedly, almost like it's responding to its environment. If you saw behavior like that, you might instinctively think, is that droplet alive? And in many ways, that's the essence of life itself. Not rigid structure, but dynamic organization. This is the core idea behind liquid-liquid phase separation, which is the physical process of biomolecular condensates. At some point in the history of the universe, matter did something unexpected. It didn't just organize, it didn't just replicate, it began to experience. But how does that happen? How does chemistry become awareness? Everything starts with physics. Electrons exist in orbitals, orbitals form bonds, bonds scale into lattices. But in biology, these lattices don't stay rigid, like in diamonds. In biology, they become dynamic. Inside cells, molecules aren't locked in place. They flow, they fluctuate, and reorganize. And one of the most important mechanisms enabling this is liquid-liquid phase separation. This is just a schematic of how liquid-liquid phase separation can organize biomolecules in little tiny water droplets that can behave in a holographic way to organize biomolecules with the environment. And we're going to talk more about how this mechanism works in future slides. Through liquid-liquid phase separation, proteins and RNA condense into liquid-like droplets. These are biomolecular condensates. They are not solid, not membrane-bound, constantly exchanging components. They form, dissolve, and reform, creating a fluid internal architecture. Instead of static structures, life uses adaptive compartments. If you would like to read more about how these adaptive compartments have played a role in evolution and the origin of life, you can read this paper. And if you want to read more about how they can mediate quantum effects in biology, you can read this paper. And the way that they do it is by dispersing or condensing, which act as on and off switches, so they can create little microcircuits inside of the cell, which can process information, similar to how our nervous system processes information. And you can remember that our nervous system is made up of neurons, which are made up of these circuits, the biomolecular condensate circuits. So this is essentially what connects everything together in biology to make it into a processing system which can couple with the environment. And a simple way of understanding liquid-liquid phase separation is almost like it's oil and water. It's the same morphology or structure that oil droplets form in water, except in biology, it's actually biomolecules, specifically intrinsically disordered proteins or nucleic acid polypeptides, which can organize themselves. Nucleic acid chains of molecules, not polypeptides or proteins, sorry about that. And these intrinsically disordered molecules can organize together into these droplet shapes, which can incorporate not only other proteins, RNA and DNA, but it can incorporate ions, pH, even temperature into the structure which allows anything to regulate it. So that means phonons, which are vibrations, can regulate these condensates, and even photons, light, can regulate these droplets if they condense or disperse. And since light and vibrational energy can regulate this substrate, that's why it can act as a substrate for consciousness. Because if you think about it, light and vibration is everything that we think and feel, whether it's imagining something or seeing something, that's photons of light, or if it's feeling something, whether it be temperature or somebody touching you, those are vibrational forces that you're feeling. So phonons and photons incorporate all of our conscious experience. And when you think about a cell, most people might think a cell is just a bag of randomly assorted molecules just flowing around, but that's not it at all. What we've come to learn is that it's highly organized down to subcellular domains outside and inside the cell, which everything is aligning together based off the structure of these liquid-liquid phase separated condensates. So again, I have a schematic here showing you how this can happen and how it can account and become awareness. So liquid-liquid phase separation creates a dynamic medium or substrate that organizes biomolecules. Molecules form a holographic information network. That just means they respond to the environment and they couple to the environment so that whatever effects come from the environment are also stored, and that information is processed not only on the surface of the droplet, but the internal organization of molecules and the configuration that those molecules have inside the droplet. And the network models the world through those interactions. So a self-sustaining loop of modeling leads to the awareness that we experience. Now scale this up. In the brain, condensates are everywhere. At synapses in dendrites around RNA and signaling proteins. They regulate neurotransmitter release, signal amplification, synaptic plasticity. This means your thoughts are not just electrical spikes. They are shaped by constantly reconfiguring molecular assemblies. The brain isn't a fixed circuit. It's a fluid network of interacting condensates, outside and inside of the cell. So you can see again how these condensates can form through liquid-liquid phase separation, and how they are adaptive in nature and they can exchange molecules and respond to the surrounding. They can incorporate ions that create the electrical potential in our nervous system. They can respond to the phonons, which are mechanical energy. They can respond to temperature. They can respond to pH. So they can respond to everything, so that every form and every force of energy can get incorporated into our experience. And this will cause them to fuse, split, grow, or dissolve. So it forms a almost invisible circuit inside the liquid substrate of all of our cells. So it has the ideal properties for information processing since it has these capacities. And this goes from molecules to mind, since our mind is made of neurons, and inside of those neurons, the biomolecular condensates organize everything in the nucleus, in the cytosol, in the organelles, near the membrane, and outside of the cell. So it enables the integration of everything, of all the different chemical, mechanical, and electrical forces that come into contact with the cell can integrate in every subcellular layer. And that's how it supports conscious awareness. So here's the deeper idea. In the traditional systems, information is stored in a specific location. But in dynamic systems like waves, information is distributed. Think about photons carrying electromagnetic waves, also phonons carrying vibrational energy. The information isn't inside a single point. It's in the pattern across the whole system. And this just shows you how the biomolecular condensates are inside and outside of all of the synapses in our brain. However, this is just a schematic. It's not an exact replication of what it looks like. If anything, I think in the brain it's even more organized into specific architectures in the brain than this is showing. But this does show how it can create these emergent patterns to transfer information. So in biology, something similar is happening. Condensates store information in their composition, process information at their interfaces, share information through continuous exchange. So every droplet is interacting with others. Every surface is a site of computation. This creates a distributed information field across the cell and across the brain. Liquid-liquid phase separation creates dynamic, semi-coherent compartments that concentrate molecules, photons, phonons, and electromagnetic fluctuations into overlapping interference patterns, allowing information to be distributed non-locally across the system in a holographic manner, where the whole organizational state is encoded within each interacting region. Information in biology is not localized. It's emergent and shared, so we can experience everything happening in our whole body. And I think this is a good schematic showing how phonons, which are mechanical vibrations, and photons can both be incorporated in the biomolecular condensate, which will organize the molecules on the surface and the configuration of molecules inside each droplet, and millions or maybe trillions of these interacting droplets in our brain all work together to process information from all our sensory organs, which gives us a conscious experience. And the reason why we have subjectivity is you can think about the trillions of molecules inside all of these millions of droplets, and you can imagine that for every person, the configuration of molecules is drastically different, even if they're experiencing similar things happening to them. Even if a person is right next to somebody experiencing a crowd of people, the molecular interactions that are going on are so different, giving this substrate that consciousness uses, that it's almost impossible to have the same experience because there's so many different interactions that can occur. And that's what gives us our own subjective experience and why there's so much variability in consciousness. So your experience feels continuous, but under the hood, everything is updating. Condensates assemble, dissolve, change their molecular makeup. Each change slightly alters the system. Each state flows into the next. This creates a temporal chain of configurations. Your perception of time emerges from continuous biomolecular updates, integrated neurosignaling, and persistent but evolving molecular states. It's not a ticking clock, but a self-updating process. So this is a good schematic showing how our perception of time is dictated by these molecular condensates, and it's all dictated by the configuration of molecules in the landscape of all the condensates that are part of this liquid-liquid phase separated mechanism. So you can go through here and read each section, and please ask me any questions from this if you have any. So where does consciousness come from? We know it depends on large-scale brain networks, integrated information, dynamic neural activity. So liquid-liquid phase separation adds another layer. A system where information is distributed, structure is fluid, and interactions are continuous. Condensates don't just support life. They enable a new kind of organization. One where matter is not static, not localized, but constantly reconfiguring itself in response to itself. And in that recursive process, matter may begin to model its own state. That's not yet a full explanation of consciousness because I think it goes deeper into the layers of the universe than even molecular and quantum states, but it is part of the bridge between chemistry and experience. So you are not a static object. You are a process, a continuous flow of molecules, interactions, and information, organized through principles like liquid-liquid phase separation. The same physics that governs simple matter scales into systems that can think about themselves. And somewhere in that transition, the universe becomes aware and you experience life. So I hope you enjoyed this one. And again, please comment and ask me any questions. And you have a good one. Have a good day.